Hi, everyone. Can you hear me?
Yes.
Oh, great. So I guess it's a good afternoon, good evening, and good morning from wherever you are coming from. So welcome to today's RNA Collaborative Seminar Series. And today we are hosting here from University of North Carolina at Chapel Hill. And we recently just formed UNC RNA Discovery Group Center. And today we are fortunate to have two great speakers from UNC. First talk is going to be presented by Dr. Christine Roden. She is a postdoc researcher at Gladfelter Lab. And our second talk is going to be presented by Aki Braceros. And she is a graduate student in Dr. Calabrese Lab. And without further ado, let's get started because I know we only have 1 hour to go forward. So Christine, go ahead.
Can everyone hear me and see my screen?
Yes.
Okay. Thank you so much for that kind introduction. Today, I'm going to tell you a little bit about my research entitled SARS-CoV-2 nucleocapsid protein-RNA interactions encode viral functions. And I am a postdoc in Amy Gladfelter's lab. So the question that I try to answer during the course of my postdoctoral work is, how do viruses perform multiple functions in the same cytoplasm with such a limited proteome? And for my model virus, SARS-CoV-2, there are about 24 proteins in total encoded by the virus. So SARS-CoV-2 has this complex life cycle involving entry on an encoding and then translation and then eventually packaging and reassembly of virions. And we think that a lot of these steps are governed by the time-dependent accumulation of this viral protein called N protein. And we think that N protein forms condensates with viral RNA to perform unique function. And in this way, the virus is able to organize its cytoplasm. So our hypothesis is that a single protein yields immiscible condensates depending on the co-condensing RNA's identity. So today I'm going to tell you a very small story that fits in with this theme about how RNA sequence encodes N protein interactions and specifies the sgRNA generation rate during discontinuous transcription. Let me see if I can. SARS-CoV-2 is unique in the way it organizes its genome in a very interesting way, in that it's broken up into these two chunks. One is ORF1AB, and the second is the structural proteins on the 3' end. And in order to generate the structural proteins, the virus needs to undergo discontinuous transcription, where they make these subgenomic or sgRNAs, each of which is translated to make a different viral protein. And we know that the recombination occurs between the TRS leader sequence, or transcription regulatory sequence, and the TRS body sequence. I've just highlighted the one for spike, the protein that the vaccine is directed against. And we know that I will tell you today that TRS-LB motifs are enriched in N protein condensation promoting features. Let's see. The number of TRS features correlates with the relative abundance of sgRNAs that are generated by the virus. And variants of concern encode mutations that may bias towards nucleocapsid sgRNA generation by creating additional motifs. And this goes a long way to explain how nidoviruses, which is the family which encompasses SARS-CoV-2, control sgRNA generation rates. So just stepping back couple slides further to try and get at the big picture question. Could viruses use condensation to organize function by controlling the RNA sequence? So what do we know as a field about how condensation is encoded in the cell? So RNA binding proteins that form condensates have common features. If we, such as RNA binding domains, canonical RNA binding domains, and intrinsically disordered regions. And it's thought that the specific recognition of RNA by these RNA binding proteins allows for the oligomerization or assembly of these multiple proteins in a single transcript. There can be further RNA-RNA interactions leading to the formation of an extensive network that leads to the formation of condensates through processes like phase separation. And we know that condensates pattern cellular transcripts. So here are some examples of cytoplasmic condensates. These can also occur in the nucleus. They include RNA transport granules and Drosophila oocyte RNA transport granules, RNP granules in the muscle. In Gladfelter Lab, we're particularly interested in this fungus, and the most popular was possibly stress granules. So I'm going to tell you today that N protein of SARS-CoV-2, which is one of the viral structural proteins, is an intrinsically disordered RNA binding protein, which has features, the features of proteins which undergo condensation, including two very highly characterized and conserved RNA binding domains and a high degree of disorder. And further, when we overexpress N protein, it forms condensates in cells by itself. And if you were to fix and perform immunofluorescence on SARS-CoV-2 infected cells, over the course of infection, N protein, shown in the bottom three panels in pink, forms these very characteristic condensates that accumulate over time. So to study how viral RNA regulates N protein condensation, I have developed a simplified reconstitution assay where I take viral RNA fragments and I mix them with recombinant N protein to form condensates. Thanks to some published work that came out of our lab several years ago now, we know that there is RNA sequence specificity that drives condensation, where some fragments of the viral RNA genome are condensate promoting, such as the very 5' end fragment, whereas others are more solubilizing, as shown by this fragment to the viral RNA genome that encodes the frame shifting region. So we know that because these sequences are identical length, that there is something about the RNA that's specifying N protein condensation. But at the time of this publication, we did not know the RNA features that were encoding this process. So this simplified reconstitution assay allows me to rapidly assess RNA protein interactions, examine the effect of RNA and protein mutations, gives both information about RNA affinity and emergent droplet properties, and finally allows for longer RNA inputs than would be used in typical MZ assays. So to make the last year and a half of my life really short into one slide, I can tell you that N protein recognizes viral RNA in two distinct ways. One is going through its second RNA binding domain in the C-terminal domain, which recognizes dsRNA independent of sequence, and this interaction encodes the temperature sensitivity of the condensation. And the second is going through the first RNA binding domain in the N-terminal domain, which actually prefers this primary sequence motif, which is enriched in TRS-LB stem loops and important for sgRNA generation. And if you are interested in reading more about this, I highly recommend you go check out my bioRxiv paper after this session. So how did we figure this out? Well, we know there's two roles for the two RNA binding domains of N protein. So we know that RNA binding domain 1 is very important for driving condensation because we have this mutation, the Y109A mutation, which it basically destroys RNA binding domain 1 affinity for RNA and completely blocks condensation of protein with purified viral RNA fragments. So what I'm showing you here is images of the condensation assay. In the green is N protein in this case. So the wild type protein is able to form these condensates with increasing amounts of RNA, but the mutant protein fails to undergo condensation. So in trying to figure out how and where the sequence that were responsible for driving RBD1 interactions for RNA, we were fortunate to be just downstairs from Kevin Weeks's lab. Kevin Weeks developed this technique called RNP-map, where we're able to mix RNA and protein in a test tube, cross-link them using this chemical called SDA and UV light, RT in sequence, and then look for mutations. Anywhere that has a mutation, as shown by these peaks in the right panel, is thought to be a site of interaction between the protein and the RNA. And what stood out to us almost immediately is the difference between this gray trace and the blue trace. So wild type protein has an intense peak right near the transcription regulatory sequence, or TRS, whereas mutant has almost completely lost the ability to cross-link specifically in this region of the RNA fragment in question. So our prediction for this is that the TRS is the primary motif that's being recognized with the N-terminal RNA binding domain 1, and that mutating the TRS should block N-protein condensation, given RBD1 mutation also blocks condensation. And that turned out to be exactly the case, whereas we take a wild type viral RNA fragment and mix it with protein, as shown on the right, we form these beautiful condensates. But if we remove this teeny weeny itty bitty stem loop, then we almost completely destroy the ability of the viral RNA to drive condensation. So what features within this RNA stem loop, this stem loop 3, are being recognized by N protein becomes the next logical question. So to answer this, I synthesized fragments of viral RNA with mutations. So starting with wild type, the deletion, as I'll remind you, completely blocks condensation. If we mutate a single nucleotide in the 1,000 nucleotides, an A to a U, we can block condensation. Similar results with another nucleotide just downstream to that, and another nucleotide just downstream to that. So the loop purine identity seems to be very important for specifying the ability to condensate. And if we are to make mutations that are just upstream or downstream of these regions, but preserve the sequence character, we see almost no effect. And further, if we mutate the A within that loop to a G, which is chemically more similar, being both purines, we see very little effect on condensation. So we know the primary sequence is very important. We next asked, does the structure of stem loop 3 regulate N protein binding and in turn condensation? And what I'll show you is that unpairing stem loop 3 enhances condensation unless this motif is destroyed. So we have the wild type hairpin under minimal condensation conditions, which is predicted to be structured, and we have shape data that suggests that it's structured at the 37. If we unpair that stem loop by just making 2 nucleotides or 3 nucleotides different in 1,000, we can see a pretty significant enhancement of condensation. And these mutations actually preserve what's called the TRS leader sequence, which I'll get into later. However, if we unpair the stem loop on the 3' side, we can see that condensation is preserved when the sequence character of the second motif, the TRS-L, is preserved. But we no longer see enhancement when we destroy the sequence character of that TRS-L motif. And this suggested to us that N utilizes a clustering approach to bind TRS-RNA, where there's a motif in the loop sequence of the TRS and a second motif contained within the stem itself, which is actually the TRS-L/B sequence. So our model is a stepwise N protein condensation with the 2 motifs on stem loop 3 or TRS, where N protein binds the loop, melts that stem loop, exposing the second site. And this allows for recruitment of additional N protein dimers to form a condensate, and co-condensation of the TRS-LB may be important for further downstream pathways utilized by the virus. So where else are there clustered motifs in the genome? So this was done with the help of Rachel Seelfun. Basically, she pulled out the density of the motif in the genome relative to position, which is shown here. We can see that there's a significant density of motifs that drive end condensation around the TRS bodies of the structural proteins, as shown by these blue arrows. So we next asked, does the local density of this YRRY motif control sgRNA generation? So to take a step back, you have to think a little bit about how you build a beta coronavirus virion. So these 4 structural proteins, spike on the outside, nucleocapsid on the inside, and a single 30,000 nucleotide-ish genomic RNA. The amount of proteins is very tightly controlled, but you can never have enough N. So we want to know, how are the canonical sgRNAs generated, what prevents non-canonical generation, and how are the ratios governed and controlled. So a productive sgRNA generation will be to make, say, members of the virion structural proteins, whereas non-productive will append 5' sequence to something like ORF1ab. So the current prevailing model in the field is that the ratio is determined by extensive base pairing between the TRS-L and B sequence. You start with a plus strand complete RNA genome, and if you want to make, let's say, spike sgRNA, you synthesize antisense RNA from the 3' side till you make the TRS-B sequence. This is able to base pair extensively with the TRS leader sequence in the 5' of the genome in a base pair scanning interaction. And this leads to completed synthesis of the chimeric TRS-L antisense sgRNA, which is used as a template to make plus strand subgenomic RNA. So when we looked at the number of identical nucleotides between model coronaviruses, SARS-CoV-2, SARS-CoV, MERS, and MHV, and we looked at the 4 individual structural proteins to assemble virions, E, N, M, and S, we found that the number of identical nucleotides, that is to say the strength of the base pairing between the TRS-L and the TRS body for each of these, was not very different. So that would lead to the question, are the sgRNAs produced in equal ratios? And this turned out to be not the case. So when we look at RNA-seq from SARS-CoV-2 infected cells generated by Kim's lab, you can see that the chimeric reads are located, most of the reads correspond to TRS-B of N, followed by the TRS-B of spike, suggesting there's this huge imbalance of sgRNAs that are generated. So how is this ratio set up? So this brings me to mechanism 2. The mechanism that I favor extensively is that N protein droplet formation and circularization are regulating sgRNA generation. So in our model, N protein preferentially condenses with the TRS-B of N over, say, spike. This leads to more frequent N sgRNA circularization and in turn generation, and less frequent S sgRNA generation, which is important for the proper stoichiometry of virion assembly. So does the number of N protein condensation promoting motifs, is that correlated with sgRNA abundance? And it turns out that that is exactly the case. So what we're looking at is the TRS-B structures for each of 7 of the structural proteins. And the red sequence is the TRS body sequence, and the green is motifs. Of course, the TRS body is encompassed within this N protein condensation promoting motifs. We can see that the TRS-B of E only has 1, whereas the TRS-B of N has 4. And further, these motifs overlap with dsRNA stem loops in the genome for the most part, and this is consistent with another feature for driving N protein condensation beyond the YRRY motif. So as I mentioned, N has the most number of motifs, which is consistent with N having the most amount of sgRNA. we next asked, what is the location of the recombination site? How degenerate is it? And how does it relate to the abundance? So our hypothesis is if more than one motif is used, this could lead to a more variable recombination site. And this in turn, again, turns out to be exactly the case, where the most abundant recombination site is usually just 5' to one of the YRY motifs as shown by this pink nucleotide. However, whereas the spike sgRNA rarely gets the recombination site wrong, this is the only off-target, let's say, recombination site of any significance. The TRS-LB of N has 4 additional highly abundant recombination sites. So N is more available in S, which suggests more than one motif may be utilized by N, consistent with N having more motifs. So finally, I will tell you a little bit about how there are mutations which create additional motifs that may enhance replication. So can we use ongoing sequencing data to mine for sequence variants which promote transmission? So we know that alpha, that variant was replaced by beta, delta, and now we're on Omicron, which is approaching the infectivity rate of measles. So each subsequent virus is more infectious. How does that work? So let me introduce you to the GSA repository of coronavirus sequencing data, where anytime you sequence a genome, you deposit in this database. And what we're looking at is a zoom in of the structural proteins in the 3' of the genome. And let me show you my favorite mutation, position 20,871, where I think this mutation alters the sequence of the sgRNA in a way that would benefit both Delta and Omicron by creating an additional motif. So as we mentioned, N sgRNA, the TRS-LB of that hairpin has 4 motifs, as originally was isolated in Wuhan in December of 2019. But Omicron has a mutation that will create a 5th motif, A to a U, and Delta has a deletion in the same position, which will also create a 5th motif in a completely different way. So these 2 mutations arose independently. And that suggests that they independently required a 5th motif. And we've tested this sequence and it enhances the affinity for N protein, and it should not alter either translation or structure. So we think that mutations who create additional binding sites in N accelerate and bias towards N sgRNA production, where if we compare the original Wuhan isolate to either Delta or Omicron, they have a higher affinity for N, which should lead to earlier sgRNA generation under limiting N protein conditions, creating more N protein and therefore a selective advantage, and then would occur normally in the Wuhan strain. So lastly, I'll take the remaining 5 minutes to talk about sorry, it looks like someone, okay. What is the function of non-TRS-YRY motifs? So they're more or less uniform across the genome, other than our notable spikes in our TRS-LB sequences. There are 813 in total, consistent with 1,000 N proteins per genome in virions. Okay, let's see. So we're able to reconstitute RNP-sized particles dictated solely by RBD1 activity. So to finally package up the genome, this 30,000 nucleotide or so piece of RNA, what you have to do is assemble N protein, about 30 or 35 clusters on a single viral RNA. And this leads to the formation of these beautiful eggs in a nest as visualized by cryo-EM in this paper. We're able to do this with 1,000 nucleotide chunks where we get, using TEM, particles which are about on the order of double the size of a particle that is in virion, suggesting that we can reconstitute a good chunk of RNP assembly cell-free in the absence of any accessory factors or crowding agents. So our model is that N protein regulates both packaging and sgRNA synthesis using a semi-degenerate RNA motif density, where early in infection in low concentrations of N, you cluster motifs in order to drive sgRNA production. And in late infection, when you have a high concentration of N, you just bind everywhere, leading to the packaging of the viral genome. In big picture, I'm interested in using, trying to learn how information is stored in RNA-protein interactions using viral RNA and protein, uncovering the RNA sequence and structure cues that specify condensation, to predict the impact of non-coding mutations on viral life cycle, design therapeutics which enhance or inhibit condensation, and repurpose RNA-encoded condensation for novel functions. And with that, I will thank my collaborators and funding sources. Please check out our paper on bioRxiv, and I will take any questions.
Oh, that's great. Thanks, Christine, for wonderful talk. So for the audience, if you have a question, so there's a Q&A part, you can enter your questions. And for panelists, Martina just told me you cannot type into the Q&A. So feel free to, put in chat or just turn on your microphone and ask a question. So, and I guess I will start. So this is really interesting. So usually it's the stem loop 3 actually controls, right? So I was wondering, does the location matter? Let's say if you take that and shuffle it to other places, will it still cause condensation? Or actually, I mean, the reason I'm asking that is, does the higher order structure matters? Because depending on location where it is.
So so one thing that I have done is appended a second TRS stem loop to the 3' side of this RNA fragment that I use the first 1,000 nucleotides. And that promotes condensation. But to get at your second point, indeed, the structure absolutely matters. The structure is important for the regulation of the binding of RBD1 to the TRS. But it's even more important in my hands for binding of the second RNA binding domain to RNA that is specifically in my hands recognizing long stretches of dsRNA independent of sequence. Yeah.
Great. Okay, so
yes.
Yeah, chat.
Can I ask a quick question?
Yeah.
I'm not sure if there's a question in the Q&A as well.
There's, but you go ahead. I'm reading the question.
Okay, cool. Yeah, Christine, great talk. I remember you saying that RNA binding protein is largely intrinsically disordered. Have you been able to find whether it has any structure once it's bound to its target?
Okay, so what has been done is you can crystallize the individual structured portions of the RNA binding domain, the N-terminal domain and the C-terminal RNA binding and dimerization domain. And there is a really beautiful co-crystal of the C-terminal RNA binding and dimerization domain with a fragment of 7 nucleotides of single-stranded RNA. And that paper is Zenzula et al. And interestingly about that domain, it sort of pushes the dimer apart, which is fun. As for the TRS co-social of that with RNA, I don't think that has been done. But there is a lovely paper that was on bioRxiv a couple months ago by a Brazilian group modeling the TRS RNA with the structure of the protein. The disordered domains themselves are not solvable, at least not yet, not even by cryo-EM. So yeah.
Thank you.
We tried.
Okay, so there are 2 questions in Q&A. The first one is from Xue Guangai, and I guess she asked, have you checked the infectivity of the condensation mutants?
On N protein or RNA would be my question. I will answer that for the mutation that I like in delta and omicron in my favorite position, we have not made Wuhan sequence with only that point mutation yet, which would be the most strong evidence. As for mutations in N protein that affect RNA binding, those have previously been demonstrated in MHV. to basically block the ability of N protein to enhance viral replication as assessed by a plaque assay. So yes, we're looking very much forward to doing that mutation.
Okay. Yeah, so another question from Sergey Shevtsov, and the question is, what is a possible signal which command N protein RNA condensate to get packed into viral?
Okay. So I think you may be alluding to something like a packaging signal, which for viruses like HIV is a very specific stem loop. That is, if you don't have that, then you don't get packaged. Coronavirus has something like that. If you've read Jennifer Doudna's science paper, she whittled it down to, I think, a 200 or 300 nucleotide fragment of viral RNA that enhances packaging. It's very similar in terms of sequence and structure to the same region in MHV, which is the BSL-2 model coronavirus. Now, if you delete that region from MHV, packaging is reduced and the selective advantage is less favored, but it's still able to package viral RNA just fine. So, but short answer is we don't really know, but it does get rid of all the other RNA in the cell and a lot of the other protein. So maybe that's just the only option around.
Okay, great. So given the time, and if you have more questions, feel free to enter your question in the Q&A, and Christine is going to address as many as she can. And now let's switch gear to our second speaker, Aki Braceros, and she's going to talk to us about lncRNA. Go ahead, Aki.
Okay. All right, can everyone see my slides?
Yes, and we can hear you too.
Perfect. All right, so first want to take a moment to thank the organizers for giving us the opportunity, specifically the UNC RNA Discovery Center, the opportunity to participate in this great seminar series. I really enjoy these virtual series because it's a great way and productive way to stay in touch with the research that's going on across the country and around the world. So I'm very excited to and honored to speak today and tell you about some of the work that we are doing in Mauro Calabrese's lab. But just as a reminder, my name is Aki and I am a graduate student in Mauro's lab. And so for my talk today, which is suggested by my title, I will be talking about how DNA regulatory elements play an important role in governing the epigenetic silencing by a very interesting long non-coding RNA called Airn. But before diving into the details of this work, I would like to mention that our lab's broad interest is to understand regulatory mechanisms of gene expression. And so one particular area that we focus on in the lab is the biological process called genomic imprinting. And so genomic imprinting is an epigenetic mechanism of gene dosage regulation that is essential for life. So here's just a simple cartoon schematic of an imprinted gene in a diploid organism, where one of the two copies of the gene is preferentially and always expressed from either the paternally inherited chromosome, like on the left, or the maternally inherited chromosome, like on the right. And as of recent, there are approximately 200 to 250, depending on the study, genes that are known to be subjected to genomic imprinting. And these genes are known to be important for things like growth and metabolism, which in turn are key, play key roles for many aspects of development. And so as you can imagine then, when improper imprinting occurs, specifically for these genes, this often leads or causes detrimental outcomes such as birth defects, developmental deficiencies, and even cancers. And I've just listed here some of the major human diseases that are caused from improper imprinting. And so one thing to note about imprinted genes is that they tend to cluster in genomic domains called imprinted domains. And imprinted domains allow for a coordinated regulation of these genes by key regulators. And so one of those key regulators being long non-coding RNAs, or lncRNAs for short. And so one example, which is the main focus of my talk and thesis work, is the imprinted domain regulated by the lncRNA called Airn. And so Airn is expressed from the paternal chromosome, specifically of chromosome 17, and it silences its neighboring genes that span across this 12 megabase domain. And so Airn silences in cis, meaning in this context means that it only acts on the chromosome from which it was transcribed. So that you'll never see it act on any other chromosome or the other allele. And so we know from our work and others that one way that Airn silences is through the recruitment of the polycomb repressive complexes to chromatin. And these are highly conserved enzymes that repress genes by compacting chromatin and depositing epigenetic silencing marks. And in regards to the properties of the Airn RNA, we know that it's incredibly long at about 90 kilobases in length, and it is transcribed by RNA polymerase II. And like most POL2 transcripts, Airn is capped and polyadenylated, but it does remain predominantly unspliced. And furthermore, Airn RNA is not super abundant in the cell. And when it is, it tends to be pretty short-lived. And so altogether, these properties in themselves cause Airn to be tethered to chromatin and to likely never leave its site of its gene locus. And just to demonstrate that, here is an RNA DNA co-fish image of a cell that expresses Airn. And you can see that when we probe or label for Airn RNA and Airn DNA separately, but in the same cell, we only get one RNA dot that co-localizes with the two DNA dots, sorry. Again, demonstrating that this RNA is tethered to chromatin. And so considering these properties about air, and we found it pretty remarkable yet odd that this unstable yet immobile immobile RNA could only mediate silencing over 12 megabases of DNA, which I want to emphasize here is over 10% of the entire length of chromosome 17. So that led us to this major question of how does an unstable chromatin tethered lncRNA like air silence multiple megabases of DNA? So we wanted to understand the silencing mechanism by Airn. So to preface, for most of our assays, we measure Airn-induced silencing by profiling the location and the intensity of the H3K27 trimethyl silencing mark deposited by polycomb through the assay called chromatin immunoprecipitation, or simply CHIP. And so in a series of experiments to see how Airn expression levels affect silencing, we used a CRISPR-Cas9 system to alter the endogenous expression of Airn. Thus, we were able to generate Airn highly expressing cells, Airn repressed or knockdown cells, as well as an Airn truncation cells that phenocopies a knockout. So then we measured Airn silencing in these cells in parallel to a wild type cell line. And we saw that the Airn repressive capacity actually scales with its RNA levels. And importantly, this RNA silencing is specific to the Airn target domain. And these data support a functional role for the non-coding RNA in silencing. So then if we look within the Airn target domain, we notice that Airn-induced silencing actually occurs in a rather non-uniform manner, where certain regions such as here, here, and here are more susceptible to silencing than others. Thus, this non-uniform pattern of silencing hinted to us that there are chromatin features contributing to this long distance and regional silencing by Airn. So then we suspected that chromatin architecture could be one of these features. And so from our own work, we found by DNA fish that the spatial distance between the Airn DNA locus and distal loci across its Airn target domain, as annotated here, actually correlate with Airn-dependent silencing. So what I mean by that is where you see a region that has high silencing by Airn, that region has a smaller spatial distance to the Airn locus. And then in a separate study by Quanah Hudson's and Florian Pauler's group, they used an orthogonal method called chromatin conformation capture, or 3C, to show that pre-existing long-range interactions actually link the Airn locus to some of its distal target genes. And so these evidence together kind of illustrate this cartoon model of what the chromatin architecture of the Airn target domain could be. And so this led to our hypothesis that chromatin architecture of the Airn target domain helps govern the long distance and non-uniform silencing by the Airn lncRNA. All right, so to address this hypothesis, we turned to a method called in-situ-Hi-C, which involves DNA-DNA proximity ligation coupled with deep sequencing, as illustrated here. And so essentially, Hi-C gives us high-resolution data to be able to quantitatively measure pairwise DNA contacts across the genome. And so with Hi-C, we're able to address some key questions. The first being, what is the chromatin conformation of the Airn silence domain, and how is that conformation influenced by Airn expression? And then more specifically, are there 3D chromatin structures and or specific regions of DNA contacts with the Airn locus that could govern or explain this Airn-mediated silencing pattern that is occurring non-uniformly? But before I get into the Hi-C data, I also want to take a moment to talk about our model system in which we perform essentially all of our assays in. And these are F1 hybrid mouse trophoblast stem cells, or TS cells for short. And so TS cells are these extra embryonic precursors of the trophoblast. And the trophoblast is a major tissue that forms a major part of the placenta. Thus, our work in TS cells actually has some implications in early developmental biology, because TS cells can serve as an ex vivo model of the trophoblast. But a main reason why we use TS cells is that they express Airn and other imprinted RNAs that our lab is interested in. And then another advantage to TS cells, specifically the ones that we use in the lab, is that they come from an F1 hybrid mouse. And this allows us to perform allele-specific analyses. And so to break that down real quick, here is our F1 hybrid mouse in which we extract our TS cells from. And it was derived from these two strains or parents of different strains. And so in this case, we have a Cas mom and a B6 dad. And the Cas and the B6 strains are known to differ by their DNA content by at least 20 million single nucleotide polymorphisms, or SNPs. And so we can exploit these SNPs to be able to trace back each allele to their parent of origin. And this actually works in our favor because, as I mentioned before, Airn is monoallelically expressed from the paternal chromosome, and it only acts in cis, so only on the chromosome from which it was transcribed. So that actually gives us an internal negative control with the maternal chromosome. All right, so for the rest of my talk, you'll see that I partitioned the data by allele, and I will include these cartoons along the data to help us keep on track on the corresponding allelic data. All right, so now moving on to the Hi-C data, I'm showing you a 2D heat map of all the pairwise contacts occurring across the 12 megabase Airn silence domain. And so to read a heat map or Hi-C heat map, each pixel on this heat map represents a contact, and the color intensity of that pixel represents the contact frequency. And so based on this orientation, you can see this diagonal line. So any pixel or contact that's closest to the diagonal line represents a short range interaction, whereas pixels farther away from this diagonal line represent a long range interaction. And so knowing this information, we can then compare the contacts on the Airn expressing allele in blue versus the non-expressing allele in red. And so knowing this information, we can clearly see already that there are differences between these two alleles. But I want to push your attention towards this area here, which is exhibiting a lot of long-range contacts. In particular, here's the Airn locus. You can see a lot of these long-range contacts aligning with the Airn locus. And if you look on the flip side, on the non-expressing allele, we don't see that same concentration of long-range contacts, particularly with the Airn locus. And so to us, this suggests that Airn expression is inducing long-range changes to chromatin architecture. But to really convince ourselves that this was due to Airn expression, we also performed Hi-C in the Airn truncation cells. And so now when Airn is absent on both alleles, these two alleles are essentially the same, or we at least lose all those long-range contacts that was on the original paternal chromosome. And so now we became interested in identifying the regions that interact with Airn. And so what I did was I extracted all the contacts that only contact the Airn locus, and then I plotted their intensities across the Airn target domain. And so that's what I'm showing you here. And so the windows here are again set to the Airn target domain, that 12 megabase region. And like before, here is the Airn locus. And so one thing I want to note about Hi-C is by the nature of the method, there's just a higher chance of detecting contacts between regions that are closer to each other in linear distance. And so this is why you see kind of like this decay effect relative to the Airn locus. But in spite of this, we can still pick out regions that contact Airn more than others. And just like we saw in the heat maps, a lot of these long-range contacts are enhanced on the N expressing allele. But still, we would like to determine the extent to which these contacts occur above what's expected based on their linear proximity. So to address this, we're able to normalize the observed contacts here by the contacts expected by chance for each loci pair. And these expected contacts can be estimated using a distance decay model. And so if we transform the data and view it now in this way, this really helps our eyes more readily pick out the specific regions that interact with the Airn locus more than others, some of which that are again enhanced by Airn expression and others that remain unchanged but are pretty prominent on both alleles. So now when we overlap the silencing data on the top with the Hi-C contacts on the bottom, we can now start to see some relationships that suggest that silencing by Airn is spreading via these contacts, sites of contacts with the Airn locus. But still, we would like to know where the Airn RNA itself is forming contacts with DNA. And so that led us to ask some key questions. What regions of DNA do Airn RNA associate with? And more specifically, does Airn RNA associate and spread silencing from sites of contact with the Airn DNA locus? So to address this, we pursued a method called CHART, which essentially works like CHIP, except it uses complementary oligonucleotides and biotinstructive chemistry to fish out your RNA of interest and the associated chromatin. And then you can just isolate the DNA from that chromatin and then analyze it as you would for chips like ChIP-seq. So then if we compare the silencing data on the left with the Airn chart data on the right from wild type TS cells, we can see that both share very similar profiles and actually correlate pretty well. But we wanted to know if DNA contacts by Airn RNA actually scales with the expression levels as observed with Airn-induced silencing. So we performed Airn chart in the Airn highly expressing cells, as well as the Airn truncation TS cells. And when we did that, we were really excited to not only see how well these experiments worked, but also to see how well correlated these independent data are from different assays are to each other. And again, these data further support a direct role for the non-coding RNA in silencing. All right, so now if we compare the data from all three assays in wild type cells, it looks like it does indeed look like that Airn RNA and Airn-induced silencing could be spreading relative to the sites of contact with the Airn locus. So you might be wondering by now, what's at these contact points? And so I can tell you that at the DNA sequence level, we observe these regulatory elements called CPG islands. And so CPG islands are these CG-rich DNA sequences that are commonly found at the promoters of genes in vertebrates. And we know that they can regulate the expression of these genes. But a highlighting feature of CPG islands in regards to our study is that they are known to bind polycomb in mammals. And the polycomb at these DNA regulatory elements can undergo long-range contact with each other to form 3D clusters that are called polycomb bodies. And polycomb bodies are meant to retain repression. So now if we look at the CPG islands that strongly contact the Airn locus, we see that one, they also overlap the promoters of target genes, specifically by air, and they too also bind polycomb as well. So that led us to this hypothesis that CpG islands form a 3D chromatin network with the Airn locus, presumably through polycomb, that is essential for Airn-mediated silencing. So to address our hypothesis, then we used a CRISPR-Cas9 system to delete 3 of these CpG islands, which we'll identify by their corresponding gene names. So we have SLC22A3, PD10A, as well as T. But we were also interested in this cluster of CTCF and cohesin binding sites between the T and PD10A CpG islands. And the reason why we're interested in them is because these 2 proteins are major architectural factors that are known to control large 3D chromatin structures that are important for gene regulation. So we decided to include this cluster as one of our target deletions to see if the regulatory elements within this area are important for Airn-mediated silencing as well. So for each target deletion, in TS cells, we obtained 2 independent clones that each harbor deletion specific to the Airn expressing allele. And then as before, we performed H3K27 trimethyl-ChIP-seq in these deletion cells to measure silencing. And so starting with the SLC22A3 deletion, we observed a remarkable 4.5 megabase reduction in Airn-mediated silencing. And we see this effect extend from the SLC22A3 deletion site all the way to the T-CpG island. And this result supports our hypothesis that these CpG islands are codependent and working together to form this 3D chromatin network that's important for Airn-induced silencing. However, when we deleted the T-CpG island on the other end, which is 5 megabases away from the Airn locus, we did not see that same effect. We essentially saw no difference in silencing, perhaps a little bit of reduction here, but nothing widespread like SLC22A3. But then when it came to deleting PD10A, which is in that same neighborhood of context as T, rather than seeing a reduction in silencing like SLC22A3, we unexpectedly saw a dramatic increase across the entire Airn target domain, except around SLC22A3. And furthermore, we even start to see a loss in that non-uniform pattern of silencing that we observed in the wild type cells. And particularly, we see that occurring over this region here that normally escapes silencing. And just really quick, we see the same phenotype observed in the cluster deletion, though to a lesser extent than the PD10A deletion. So based on these deletion results, we've illustrated what we think is going on with the chromatin architecture of the Airn target domain. So our initial high-C data suggests that these CpG islands are frequently interacting the Airn locus. But specifically, we think that the SLC22A3 CpG island kind of acts as an anchor that bridges the Airn locus to these other CpG islands, which explains the codependency of these elements and the multi-megabase reduction that was observed in the silencing caused by the deletion. And then as for PD10A, it appears to play a critical role in promoting a second architectural state that helps these regions escape full silencing by Airn. So we think that there's kind of like an equilibrium between these 2 states. Thus, when you delete the PD10A, it may cause a collapse in these 3D DNA structures that then become more vulnerable to silencing by Airn. And this was definitely the most surprising result, given that we initially just looked at PD10A as just a silenced target by air. But now we're appreciating more that through some RNA-seq data that PD10A gene is actually only mildly repressed by Airn and actually exhibits a more what's called a bivalent chromatin state, which just means that it's poised to be active or silenced. All right, so to summarize my talk, I showed you that Airn expression induces longer range changes to chromatin architecture that center around a network of CpG islands. I also showed you that DNA sites of contact by eRNA and Airn-induced silencing are highly correlated, and these likely are directed by the CpG islands. And then lastly, we saw that deletions of these DNA regulatory elements, particularly the CPG islands actually cause a widespread yet variable changes in Airn-induced silencing. So I want to finish off with some broad implications that our work demonstrates. The first being that DNA regulatory elements and non-coding RNAs work in concert to regulate gene expression, for example, in genomic imprinting. And so some follow-up questions to that is, what features of DNA elements and non-coding RNAs give them their functions? What is it about their sequence? What is it about the protein factors that bind them? And this work also inspires the idea of that there are other chromatin-bound RNAs that likely work through DNA regulatory elements to control other regions of the genome. And so considering these relationships between RNA and DNA, understanding how they work together could give us a better handle on controlling gene expression in specific regions of the genome, particularly in dysregulated regions that actually cause some of the diseases that I've talked about. And so with that, I just want to quickly acknowledge the members of my lab, both present and past, as well as our collaborators, core facilities, thesis committee, and the funding. And with that, I will take any questions, and thank you again for tuning in.
That's great talk, Aki. So I already see there's one question in Q&A, so I'm going to read that in the time. Sergey Shevtsov asked another question. He said, I think you should use fish to show that your CRISPR deletions actually do anything with the 3D organization of those loci, even before you start checking what is happening with the regulation.
Yeah, yes, I agree. We are kind of like on the fence of performing FISH or Hi-C or some variation of Hi-C to be able to look at the actual chromatin architecture and see what these deletions actually cause. So that's actually definitely a very big interest of ours to do next.
Great. Any question from the panel? So actually, this is actually quite amazing. So can I ask a naive question? So you mentioned Airn has a very short lifetime, like an hour, but it's able to maintain just 8 copies. So how does it do it? I mean, is the one destroyed and then it immediately make another one, or I mean, how does that work?
Yeah, and essentially we think it's acting as it's being transcribed, because if you consider like the rate of transcription by polymerase by Pol2, like it's like as soon it doesn't even get to be completely finished before it can even move away. So that's why we think one reason why it's being tethered to its site of transcription. But yeah, we just think that as soon as it's being transcribed, it's already acting. And we kind of have an idea that a lot of the proteins that give Airn its function are binding closer to like the 5' end of the RNA. And so at that point, like it's able to already recruit RNA binding proteins earlier as it's being transcribed. And so by the time like it does its silencing action or recruitment or whatever, it can just be degraded.
Okay, great. So there's one more question in Q&A. So it's by Anguli and great talk. Are there trans binding to other chromosomes? If so, would it also contribute to K23 trimethyl changes in those regions?
That's a very great question. So we saw something pretty interesting actually with the deletions is when we, for the PD10A, for example, deletion, we actually see a reduction in H3K27 trimethyl, the mark in like the Hox cluster. So there is some trans effects that's being caused by at least these deletions. We don't really see changes by when we alter the expression of the RNA with this mark in other regions, but with this deletion, we are seeing changes in the mark in things like the Hox cluster. We are very interested in looking or trying to address the question of whether there's trans or interchromosomal interactions that could be playing a role in all of this. So I really enjoy that question.
Great. And I think Sergey has a comment. Usually non-coding RNA don't move far away from their side of transcription. Yeah, okay, so any other questions? So sorry, Aki, I'm just trying to show one slide here. While people are still asking questions, feel free to put your question in Q&A, and for panelists, feel free to ask by turn on your unmute yourself. Here, I just want to make one advertisement is the North Carolina RNA Society is going to host a 2022 symposium on RNA biology, RNA tools, and target meeting, and it is hosted by UNC RNA Discovery Center, headed by Dr. Chad Packard, in collaboration with the Duke Center of RNA Biology, as well as NIHS. And we are featuring 3 keynote speakers and a lot of invite speakers. And if you are interested, feel free to check out our website, ncRNA.web.unc.edu, and the registration is open. And if you are traveling from outside state of North Carolina, come to here, we do provide travel awards to students and the postdocs. Okay, so Aki, you can turn the camera on, and if there are any more questions. Oh, I see there are in chats. Yeah, if no, let all thank our 2 great speakers, Christine Roden and Aki Braceros, for 2 wonderful talks. And thank you all for joining us.